Our next speaker will be Pamela Bjorkman. She is the Max Delbert Professor of Biology and an investigator of the Howard Hughes Medical Institute at Caltech. She's widely recognized as a pioneer in the field of crystallography, and much of her research has centered on the study of the structure function of MHC class proteins and other proteins of the immune system. She received the Gairdner Foundation International Award with Don Wiley in 1994 and has also been recognized with membership in the U.S. National Academy of Sciences, and more recently won the L'Oreal UNESCO Women in Science Award. Today, Dr. Bjorkman will tell us about engineering improved anti-HIV antibodies using structure-based design.
Okay, so today I wanted to tell you about our fairly recent efforts on working on HIV, and specifically to try to improve the types of antibodies that people get in response to HIV infection. So I think you're all aware that HIV is a retrovirus, and this is sort of a schematic of its life cycle. So it comes into the cell through binding to its host receptor, which is CD4, host protein on usually T cells, and then through binding to CD4 and then the co-receptor, a chemokine receptor, that allows fusion of the envelope glycoprotein that's on the virus. It gets into the cell, reverse transcribes its RNA genome, and then that gets integrated into the nucleus of the cell. And then when it wants to make more virus, it transcribes and translates its proteins and buds out of the cell. Goes on to infect a new cell. So there are many highly effective small molecule drugs that are being used across the developed world to prevent HIV transmission to an infected person, and they affect different parts of the life cycle. You can inhibit the initial entrance, a lot of reverse transcriptase inhibitors, there are integrase inhibitors, and there are HIV protease inhibitors to inhibit the maturation of the polyprotein of HIV. Now, if those work quite well for infected people, but the problem is that for a variety of reasons, they're not working globally. So we still have 34 million people living with HIV infection across the world, and it's particularly bad in sub-Saharan Africa. So something needs to be done because the small molecule drugs, just for a variety of reasons, aren't working in the developing world. So the best thing that the whole world could have would be a vaccine against HIV. So a conventional vaccine would work by injecting something, and that might be attenuated virus or whatever. So here's the HIV with its envelope spike proteins, and it would be injected somehow, and you would raise antibodies against it. That's how most vaccines work, by the antibody response. And you'd raise a variety of antibodies. You'd neutralize the virus before it could even infect. And the problem with that is that there's no effective vaccine despite 20 years or more of trying. So, and there are a variety of reasons for this. And one of them is that HIV has evolved a lot of ways of evading antibodies, so that making what are called broadly neutralizing antibodies against HIV is very rare. So this is a schematic of the envelope spike glycoprotein. It is a trimer. This is from cryo-EM studies from Sriram Subramaniam's laboratory. And you can see right here, there are a number of reasons that it's difficult for antibodies to be effective against this envelope spike. First of all, the virus rapidly mutates, so the spike keeps changing. This is from error-prone reverse transcription. There are conserved regions, but they're mainly hidden at the trimer interfaces. And then there's a lot of host-derived glycans on this protein. That creates a sort of a shield, because glycans are normally non-immunogenic because they're made by the host cell machinery. And so for a variety of reasons here, it's very difficult to raise what are called broadly neutralizing antibodies. And what I mean by that is that this is the only target of antibodies on the surface of the virus. So basically there's this spike glycoprotein, and that's it. And so when you raise antibodies, that the host does that, but they're strain specific. So they're specific to whatever strain elicited them, and they don't tend to work with the mutants that arise through evolution in the host itself. But over the years, a variety of broadly neutralizing antibodies have been isolated through single-cell cloning techniques, especially in the past 2 or 3 years. And these are isolated, though, from rare individuals. So from, say, 5 to 10% of HIV-infected people will make broadly neutralizing antibodies. But in those cases, it takes 2 to 3 years for them to make those antibodies. So it's a rare event to make them. And we started thinking that maybe what could be done, instead of trying to do a conventional vaccine approach, would be to deliver already isolated broadly neutralizing antibodies to people therapeutically. And this could be done either by injection, so this would be just passively injecting a purified human-derived monoclonal antibody into people, or a mixture of antibodies, or directing their expression through various gene therapy efforts in a person who was at risk for developing HIV infection. And so I won't go into how that might be done, but a variety of people, including Phil Johnson at UPenn, the Vaccine Research Center, and David Baltimore at Caltech, are working on gene therapy ways to deliver these broadly neutralizing antibodies. But what we wanted to do was develop more effective broadly neutralizing antibodies that could be delivered in some way. So when we started this, we thought, okay, we'll just make them into more avid forms of antibody, because a normal antibody has 2 arms to it. It has 2 antigen binding fragments, or Fabs, but the immune system makes antibodies that are more valent, that have more valency than just bivalency. So it makes tetravalent IgA, this is a dimeric IgA, and it makes IgM that has either 10 or 12 Fabs. And so first we took the existing broadly neutralizing antibodies that were IgGs with 2 Fabs, we converted them to IgAs, IgMs, they didn't work any better. And then we converted them to all kinds of designs that don't exist in nature. We thought we could make better antibodies through design. And about 100 or 200 of these constructs later, we had nothing that worked any better. So we started thinking that maybe the virus had more tricks up its sleeve than we understood, because none of these things were working. And we came up with this idea that actually there was another reason that hadn't been appreciated for how HIV was managing to elude most antibodies, and that is that it has a very low density of targets for the antibodies on its surface. And so let me show you this pictorially. These are images of different viruses, and the first 4, we have either vaccines against them to raise antibodies that are effective in protecting a person, or the body makes its own effective antibody response to clear the viral infection. And what you can see is a dense array of envelope spikes on these viruses. And now you look at HIV. This is from cryo-electron tomography from Kenneth Rue's lab, but it's also from the Subramaniam lab. So when you look at individual HIV particles like this, they look strange. Well, they basically look bald. So what they have is an average of only 14 spikes, so maybe up to 20, but most of them have under 20 spikes. And these are sparsely distributed on the envelope surface. And by comparison, HIV is about the same size as influenza virus, so 100, 140 nanometers or so in diameter. Flu has 440 spikes, HIV has an average of 14. And so why does this matter? Well, if you look from the Rue labs, they did a nearest neighbor distribution histogram of the average distance between spikes on, say, 40 or 50 viruses. They plotted this. And most of the spikes are, far enough apart that there's a 15 nanometer distance here, which is the average distance between the arms of an IgG antibody. So this is where it would bind to antigen. And most of the spikes are further apart than 15 nanometers. So most of the time, you're not going to have bivalent binding between spikes. So so-called interspike binding, where a Fab binds to this spike and another Fab binds to this spike, will not occur. And this is important because this allows the antibody to bind with avidity, so that if it's binding with 2 spikes, as the mutation happens, the affinity of an individual arm for the antigen will go down because the spike is actually changed. Now, if you have avidity going for you, you can accept some of this mutation, so that what we're trying to say here is that one of the reasons that HIV is able to evade antibodies is that there's a low spike density, and if you combine that with the rapid mutation that HIV is capable of, that creates another reason it's a deadly combination here, because there's no so-called buffering against the effects of the mutation. So in other words, if you didn't have the mutation here and you bound with one Fab, you'd be fine. It would bind with high affinity. But as soon as you throw in the mutation here and the affinity of that Fab goes down, you don't have any buffer against it by being able to bind with two arms at once. So we thought that in general, this created a way to make better antibodies, those that could actually bind bivalently. And the way you can get around the fact that there aren't very many spikes and they're distributed at large distances would be to do so-called intraspike crosslinking, so bind with both arms to the same spike. And that you should be able to accomplish this in theory because these are trimers. So there would be, the same epitope would be repeated 3 times on a trimer. Well, a normal antibody, due to the geometry of this interaction, this doesn't happen for most epitopes on the HIV spike trimer. You can show this through modeling of the approximate antigen binding sites here. It's not going to happen. But maybe you could make something that had a longer hinge region. Or you could, if you were doing something totally unnatural, you could make a trimerized version that had 3 binding sites. Okay, so then we started making these sorts of constructs to try to get intraspike cross-linking. And again, 100 or so of these, and we couldn't find any that really worked much better than their original starting parental molecules. So what we decided to do was to try to develop a way to measure the distance between the individual antigen binding sites. And this is a project that we're ongoing now, and what I wanted to show you is we have some promising results for doing this. So what we're trying to do here is, because there isn't a high-resolution structure yet of the HIV spike trimer, rumor has it it's coming, a 4.7 angstrom structure is coming out of a trimer from Ian Wilson's lab in a couple of weeks, but even that won't tell us where the antibodies all bind. So what we're trying to do is measure that distance. So we're using double-stranded DNA as a sort of molecular ruler that has, as you may know, a relatively long persistence length that's different than extended polypeptide chains of proteins. So up to about 50 nanometers, it behaves as a relatively rigid rod that has a 3.4 angstrom per base pair spacing between the base pairs. So the idea is you take antibody Fabs that bind to HIV, you conjugate them to different lengths of double-stranded DNA. You then assay these to see if they do intraspike cross-linking, and I'll show you the assay we're doing with these libraries of Fabs is an in vitro neutralization assay of HIV entry into target cells. And then once you get the ideal distance between the Fabs, you would know that distance, and then this is critical, you would convert that to a protein-based linker, because what we're trying to do would eventually be encode something with a gene, so it has to be an all-protein reagent, and plus if you actually injected this into someone, you'd have immune reactions. Okay, so I wanted to show you that we think we're on the right track here with just one example. So here is a case where what Rachel Galamidi has done is to link an antibody called PG16, it's against one of the variable loops on HIV at the top, to a CD4 binding site antibody called B12, and she links it with a linker of about 60 base pairs, and these two are linked to each other, and now she assays them against 2 different viral strains. One of these is sensitive to B12, so it neutralizes relatively well at a relatively low concentration. These are the concentration of the potential inhibitor, and this is the % neutralization measured in vitro to a reporter cell. Okay, so B12 works relatively well. PG16 doesn't work so well on this strain, and this strain is the opposite. PG16 works well, but B12 doesn't work so well. And now what you do is you take the so-called heterodifab, which has PG16 and B12 covalently linked to each other via this ruler thing, and you can see that this works much better. So what we're achieving here, and this is after hundreds of assays where we did not achieve this, so this is, I won't show you all the negative results, but what we're achieving is synergy. And so what that would say is that it's not that B12 or PG16 is binding. That's easy to make. We can make lots of reagents where one or the other is binding. This means that both are binding. We think this means that both are binding at once to achieve bivalency, to have an avidity effect, and to allow both epitopes to be occupied at once. So this, in both cases, you see a lower potency or a better potency of neutralization. And this is the control. You just mix PG16 and B12 with the double-stranded DNA, but they're not covalently linked. And now that mixture is no better than the best of the reagents there. So we think we're on the right track to identifying how to make a reagent that will bind multivalently. So this is our ideal reagent. And we have a challenge to HIV, which is try to mutate from this, because I don't think it can. So the idea is this would be the ideal reagent. It would be something that in a single arm is able to bind to 2 or more different epitopes on the same monomer of the trimer. And then we would multimerize that so that you'd be binding to either, it's as depicted here, say 6 places, or if you had 3 places per trimer, we're working on those too, you'd bind to 9 different places. And I don't think that HIV could actually mutate fast enough. So the principle of highly active antiretroviral therapy, which is what's given to people today, is that they take, say, 3 different anti-HIV drugs, and the virus can't mutate fast enough to make mutations in a cycle that would overcome all 3 of those drugs. And so by the same token, if we had 2 or 3 different epitopes here, the virus couldn't mutate fast enough to get around them, but we'd also have the very strong avidity effect here, so that even if the virus did mutate in 1 or more of the antigen binding sites, avidity effects would allow the reagent to still bind. So this is what we're working towards. And I want to switch now to what I said I was going to talk about, which is using structure-based design. So this would be a more rational type of approach that's done by actually looking at individual structures. And here what we're trying to do is improve the individual Fab of an antibody so that it's more broadly, so that the antibody itself is more broadly neutralizing. And these could be, if we can do these improvements, these could be used as conventional IgGs. They probably won't bind bivalently, but maybe we can improve them enough to be used in that sense. Or we can take the improved Fab and use them in the previous approach. And this is a collaboration with Michel Nussensvag and his lab at Rockefeller University. They have isolated hundreds in the past 2 or 3 years. They've done single-cell cloning techniques, getting B cells from HIV-infected donors, and then isolated a huge number of new and very exciting broadly neutralizing antibodies from these rare subset of people, the 5 to 10% of HIV-infected people who make broadly neutralizing antibodies. Okay, so as part of our collaboration with him, we're trying to figure out the structural correlates of broad and potent neutralization of HIV. And for that, it's important to have a lot of different antibodies so that we have closely related relatives. Some neutralize really well, some neutralize medium well, and some don't neutralize very well at all. And we need to have these comparisons of the bad ones, the medium ones, and the good ones to say, what is actually going on here to make something a broad and potent neutralizer. So here's one of our first crystal structures in collaboration with Michel. This is an antibody they isolated from patient 45, and they called it NIH 4546. And it's a more potent clonal variant of an antibody isolated the previous year from the Vaccine Research Center, which was called VRC01. So VRC01 was the first very broad and potent neutralizing antibody against the CD4 binding site on HIV GP120. That's one of the subunits of the spike trimer. So this is a crystal structure that Ron Diskin in my lab did of, there's GP120, that's the HIV protein, and here are the 2 chains of the Fab. And the details here aren't important about what's going on, except to say that because 4546, Michelle's antibody, was more potent than VRC01, but it was 86% identical in its heavy chain, and even more than that in its light chain, we were able to ask what part of it makes it more potent. The answer was an insertion in the 3rd hypervariable region of the antibody. That insertion is right here, so we knew that from grafting experiments onto both of the antibodies. So that's the reason it's more potent, and that's structurally the reason right there. Okay, so this was interesting, and it suggested what you could do to maybe improve other antibodies. But Ron Diskin wanted to go further and see if he could improve 4546, which was already incredibly good. And so to think about this, I'm going to back up a little bit to remind you that antibodies already undergo evolution to improve their affinity and their ability to bind to their antigen. And this is the process of somatic hypermutation, in which your first response to an antigen, you get B cells that have B cell receptors. Those are membrane-bound antibodies. They bind with relatively low affinity to the antigen. You get a bunch of those different types here. But now the B cell goes back to the germinal center, sees the antigen again, it undergoes hypermutation of its variable regions, and the ones that bind better to the antigen come out and make better antibodies, and you can have several rounds of this to get high affinity. Well, in the HIV case, it's really interesting because the average number of somatic hypermutations in other antibodies and other systems is, 10, maybe 15 at most, but 10 would be more typical. And you can have up to 60 in these broad and potent HIV antibodies, which is really amazing. And it actually suggests to people, it's one of the reasons that the field wants to develop, say, gene therapy as a way of delivering the antibodies, because if you have to make 60 mutations in an antibody to make it broad and potent, how in the world are you going to immunize to get that if it takes 2 or 3 years for the individual to develop that type of broad and potent neutralization response? So that's an aside. But anyway, if you think about that, maybe these CD4 binding site antibodies that have such excessive somatic hypermutation, maybe they're optimally mutated as they are isolated from the patient. But Ron didn't think so, and that's fortunate, because what he did was to look, and this is shown in a movie here, he looked at how this antibody was mimicking CD4. So it's a CD4 binding site antibody, and CD4, the structure of CD4 bound to GP120 is in red, and the structure of the heavy chain of our antibody is in blue. And GP120 is this gray surface right here. So GP120 has a conserved hydrophobic little tiny pocket on it that accommodates phenylalanine from CD4. And that's critical for CD4's interaction to recognize GP120 and be the host cell receptor. So that phenylalanine is critical. But what NIH 4546, the antibody imitates it, you can see quite well, but it has a glycine at this position. And its clonal relatives, such as VRC01, also have a glycine. So they have no side chain at all here. So they're not putting anything into this pocket. So what Ron did was to make a series of hydrophobic substitutions at this position in this antibody, and that position is 54. And so eventually he made a very, very good antibody that's called, we call it G54W, which means he substituted the glycine at this position 54 of the antibody for a tryptophan. And he made other substitutions that are also good, but this one was the best one. So I'm going to show you on this coverage curve a summary of the efficacy of that antibody compared to a variety of other antibodies. Okay, so this is a coverage curve where what you plot is the percent of viral strains of HIV that are neutralized as a function of the concentration of the antibody, the concentration at which the antibody achieves 50% neutralization. So this is on maybe 100 to 200 strains of HIV that these are tested on in vitro. Okay, so here's the coverage curve for VRC01, previously the best broadly neutralizing antibody out there. And so what you want to have is 100% neutralization of 100% of the strains. Nobody's there yet, but that would be optimal. And then you want to be as far over to the left as possible, so you want it to work at really low concentrations. So there's VRC01, and there's the parent NIH 4546 in red right here. And then here's the single substitution of that antibody, G54W. And so this has been suggested as a cutoff for the optimal IC50 to be actually therapeutically effective. And so you can see that the improved antibody hits 74% of the strains at this concentration, whereas the parental antibody hits 43%. And then the less potent clonal variant, VRC01, gets 27% of the strains. So this is pretty good. But we wanted to go on and make variants of this. And so to do this, Anthony West in the lab developed a bioinformatic software tool. He calls it Antibody Database, and it's freely available to the community. It's published, oh, that paper's actually published. It's in PNAS. I think it came out like a month or two ago. So the software is available to people for doing this. But what he did was to look at all the sequences of strains of HIV that either were or were not neutralized by these antibodies. And these are available from the Los Alamos database. There's like 3,000 or 4,000 of these sequences. And then he took all the sequences of CD4 binding site antibodies that are related to this antibody, and he came up with a motif that explains which strains are not neutralized by this antibody. And the motif, we call it the asparagine or aspartate, and then asparagine, glycine, glycine motif. And this is schematically showing how the Fab, which is in magenta, interacts with GP120, which is in black. So again, the details aren't important, but the point is that there are 2 asparagines in a row on a certain loop on GP120, and there are 2 glycines in a row on a certain loop, on another loop on GP120. And if the virus mutates in either of these positions, Anthony predicted, you could escape from this antibody. So what to do about that? Well, what Ron did was he went and made 2 further mutations. These are 4546 with the W in it. It's got the tryptophan in it, but he made 2 additional mutations. The first one, so it's got G54W. The first one is a serine to a tyrosine mutation on the light chain. It increases in interaction with a carbohydrate on the GP120. And then the second one that's shown schematically right here. This is the glycan site. This is the light chain of the antibody. And then the other one was specifically designed to prevent the virus from being able to mutate in that GG part of the NNGG motif. So there's a conserved set of tryptophans at the center of the antibody. The antibody here is shown in magenta. And then these glycines right here, they have to be glycines. If the virus mutates to put in a larger residue, and any residue would be larger than a glycine, then the antibody no longer works because there isn't room. Okay, so that's the reason for this that's the reason for this route of escape. So what he did was to substitute this tryptophan here for a valine, a smaller hydrophobic residue. And then he tested to see if these mutants would be better in single amino acid changed viruses. So this is our starting antibody right here being tested against a panel of 6 viruses. And so you start and you measure the IC50 values, and you can see that when you make single amino acid changes in this motif, so the motif is N at position 279 or 280, and then GG. So these are deliberately introduced into viruses, and then we test them. And so if you're not neutralizing, you're white, and then if you are neutralizing really well, you're in blue, and then these are intermediate. So that's the parental starting one, and these are the results for the 2 mutants. We've converted them to 2 antibodies that will neutralize across these difficult to neutralize strains. And then if you look across the whole panel of strains, the 100 or so strains we commonly test against, the M2 mutant here has really improved its neutralization coverage. It may not look like much, but we've got it to within, say, 96, 97%. It's getting that many of the strains, which is really quite remarkable. So we think we're almost there. Okay, so now is this route of mutation really, does it really matter? So what Michelle Nissenschwag's lab did was to introduce NIH 4546W into mice that had a humanized immune system and had been infected with HIV. So they have human T cells, they can be infected with HIV, and then he looked at the mutations that come in response to the presence of that antibody. So he takes out the viruses that are able to grow in the presence of that antibody, and he looks at where they're mutated, and the viruses, sure enough, were mutated at N, G, or G. I'll come back to this mutation again. So that confirmed Anthony's predictions, but now what about the new, the new mutants? This is the original parent one, and that's N, G, or G. Okay, so now he does the same experiment, but he treats them with 4546, these new mutants right here, and he has completely eliminated this route of escape, exactly as designed. So the viruses can no longer use that route, but they've picked up another mutation right here, which is outside of his motif, but it turns out that that mutation introduces a potential N-link glycosylation site at position 279, so it really is part of that motif after all. And then more interestingly, that particular mutant, when you look at its in vitro fitness, in an in vitro fitness assay done by Leo Stamatatis' lab, when you look at that mutant is less fit than other mutants and from this parental virus. So we think we've forced the virus in the presence of this new designed antibody into a less fit state. And that's almost there, because if we could use antibodies that maybe the virus can't completely eliminate its route of escapes from, we don't know yet what to do about the mutations in the NN part of the motif, but if we can force it into a route of mutation that makes it less fit, then we might be part of the way there to eradicating it. Okay, so I wanted to acknowledge this has been a great collaboration with Michelle Nissenschwag's laboratory. And the people in my lab who've been doing this are Ron Diskin, who's now running his own lab at the Weizmann Institute. Rachel, I talked about earlier about the DNA Fab project, and I didn't have a chance to talk about the rest of these three people here, but Anthony West did this antibody database program. Thank you.